All right, this is a quick overview of everything in Unit 6. This isn't all the detail that we need, but this is the major points. So in Unit 6, we're talking about ecology and also population change. In ecology, we're talking about individual organisms, populations, what makes a species, a community, look at an ecosystem, look at a biome, look at the whole biosphere. So for individual organisms, the things that you need to survive are the things that you can think of that you need to survive. Whether you are a bacterium, whether you're a lichen, whether you're a whale, or whether you're an ant or a human or whatever, you need to have water, you need to have food, you need to have a place to shelter, you need to have the right temperatures, you need to have the right gases around you, you need to have the right pH. All that stuff is necessary. Those things, most of these are, that I've listed, were abiotic factors. Things like water, things like temperature, things like pH, oxygen. Those are all abiotic factors. When we're defining species, the definition we're using in Bio 20, which is definitely not the only definition for species, but it is the one everybody starts with, so we're going to start there. The definition we're going to use is if you have an organism A and organism B, can they mate and make offspring? If yes, we're halfway there. But if that one, so we take another A and another B, if they mate, they have offspring too. Maybe I'll make that one D. Can these two mate and have offspring? So are A and B in the same species? The question is, can we get to here? Can we have grandchildren? Grand mules, grand whatever. And the answer is not always obvious. In most cases, if A and B are not in the same species, then we never get here. We never get any kind of offspring. If you take a giraffe and, I don't know, sunflower, of course there's never going to be any kind of giraffe-sunflower hybrid. But sometimes you can get C, like in the case of a horse and a donkey making a mule, or a lion and a tiger making a liger or tigon. But that is not always a fertile offspring. It's not always able to make gametes that are going to be able to fertilize. So the test is, can you make grandchildren? Our definition of population and community: population is same species, same place, same time. Community is every species in the same place at the same time. At an ecosystem level, we look at all the communities, and also we include the abiotic components. What's the temperature? Is there enough rainfall? What are the nutrients in the soil? That kind of thing. The biosphere includes all of the ecosystems in the world. The biosphere just means the Earth, but biosphere sounds so much fancier. Climate is the main factor that affects which biome we're in. Biome is like a really big ecosystem, big style of ecosystem. Climate is mostly determined by how far away from the equator you are. Inside of that region, we're also concerned with what are the temperatures like, is there enough sunlight, and is there enough liquid water? Liquid water, I'm specifying here because all life needs liquid water, as far as we know, to do its stuff. Solid water, ice, not good enough. It's not good at dissolving things because it's a solid. Gaseous water, it's too hot. It damages all the stuff, the macromolecules we need to stay alive. We've looked at habitat versus range, I think, in class by now, who knows? And habitat is the part of the area where something lives, where it can find the things that it needs to survive. It can find the food, the shelter, the mates, etc., that it wants to be able to survive. If you were to plot out where you have found that organism actually surviving in nature on a map, you would be describing its range. So in the range of the giraffe, not every place will actually accommodate a giraffe. The habitat for the giraffe has to be someplace where a giraffe can find food, where it can walk around. Inside of the range for a giraffe, you may find mountains, and giraffe aren't very good with mountains. You might find lakes. They're not very good swimmers, to my knowledge anyway. And you have to be more specific inside of that range to find the exact place where that giraffe might be happily surviving. Similarly, if you find the right habitat, that doesn't necessarily mean you're going to find that organism. So if you find a place that you think, wow, this is a really good spot for a... it doesn't mean it could live there. If you find a really, really good spot for an alpaca, which is kind of like a llama, that doesn't mean that an alpaca is living there. It just means that if there were alpaca there, they could survive. Because we're not in the range of the alpaca, then if we look around Alberta for places where alpaca might survive, we're not going to find them, even though they could. So both of those criteria have to be met. We have to be inside the proper range, and inside of that range, we have to find the right conditions. And for those of you who are going to be thinking about alpaca farming in Alberta, just put that aside. Farms are not fitting into this discussion really well. Inside of your habitat, there are many niches, many different hiding spots, many different spots with slightly different conditions. The more niches you have, the more species can survive. A measure of the number of species that can survive in an area, or that do survive in an area, is called its biodiversity. In order for two species to stay in one place at the same time, at least for a long time, for an extended period, they have to be exploiting different niches. They have to be eating different food, or maybe they have to be sleeping in different places, or maybe they have to be active at different times. Something has to be different. Biodiversity is greater in places that are warmer and wetter. We have higher biodiversity in places in the tropics than we do in the Arctic, for example. For life to exist, we have to have all the conditions for life met. And whichever one you run out of first is called your limiting factor. Whatever you are limited in your supply of. So if you have all of the water in the world and all of the food in the world, but it's a 2 square foot space, I don't care how many humans could survive on that amount of water and food. Only one human will fit in that spot. Limiting factors can be abiotic, like amount of space or amount of water or sunlight or whatever. Or they could be biotic. Biotic can be something that is intra-specific. That means within the same species. Intra-specific means you're competing for food with members of your own species. If you are an ostrich, you eat ostrich stuff, and so do other ostriches. So the more ostriches are around, the more competition there is for food. Same thing for mates. If you want to reproduce, you need to have a mate, and if there is none for you, then that is something that you must compete with your own species for. Water, whatever. The things that you need are the things that the members of your own species need, so this competition can be pretty great. The other type of biotic limiting factor is interspecific. That means within different species. And that can be the same as intraspecific things. You're both competing for food. Sure, lots of different herbivores will want to eat the same grasses or the same leaves or whatever. But it can also be different. It could be predation. You could be competing, you could be a rabbit and competing for grasses with other rabbits and also with other herbivores, but you could also be hunted by fox or coyote or whatever. Same thing for parasitism. You may have your population limited because of disease. If disease spreads through an area, this is not an area where a lot of you can survive. Whether that's a bacterial infection or a parasite or what. If we're trying to measure how much biodiversity there is in an area, or just for one species, how many there are in an area, it is rarely the case that we can actually count them all, because the world is big and we are easily bored. We run out of time, we run out of money. So the idea is we take a small sample and we extrapolate. So we notice that if we're looking at this as a graph for number of organisms we see over area, we notice that we see this many, this many, this many, this many, and we're done measuring, but we really want to know what happens up here. Well, we would draw a line and we would say, okay, if we get to this point, if there's this much area, there should be this many organisms. So we take the data points we actually know about and we extrapolate a line out until we can guess what the true population is. The two techniques, one is a quadrat, where we take a grid. Usually this grid is something you can hold in your hands. You lay it down on top of the grass or on top of a bush or whatever, or even on the ocean floor. And you get in real close and you look. You're counting the number of ants you see, or counting the number of blades of grass you see, or whatever. And if each of these grid spaces is maybe 1 square foot or 1 square decimeter, then you can get the average population per square foot or per square decimeter or per square centimeter, and then extrapolate out to the area you're trying to figure out about. So if you're looking at a huge area and you really only have space for a grid this big, you'd count how many fit in one of these squares, and you'd say, okay, how many of those squares fit into this big space? And you just multiply it out. That's good for really small things. The second technique is called a transect, where you're going to take a straight line walk. So you have a big forest. Here's the zoom satellite view of the forest. Here's maybe a river running through it, I don't know. You're going to take a straight line walk through one tiny bit. And you're going to look off to the side and you're going to count how many things you see. You're going to say, okay, that makes a rectangle this big. How many of those rectangles fit into this big space? So how many similar sized rectangles could we fit into here? And you'd multiply it out. So this is really good for things that are big. This is really good for things that are small. Both of them are really only great for things that stay still. Because if you're trying to count the number of squirrels, by the time you lay a grid over top, the squirrel is gone. And if you're taking a walk through the forest, the squirrel is probably also going to go hide. If you're looking for birds or whatever, same problem. So sampling has many different techniques. Here are just two of the major ones. Naming things in biology is a major deal. It's a whole field. Taxonomy is the science of splitting things into groups called taxa. A taxa, or is plural, and taxon is singular, and taxon means just a group. So if we're splitting things into different taxa, we're trying to categorize them, we're trying to make names. We're using a system that is developed from the system made by Linnaeus. And Linnaeus wasn't born Linnaeus, but we'll talk about that maybe in class. And the Linnaean classification system has been tweaked and changed and is continuing to tweak and change over the years. But the broadest category here, the way that we start to look at it, is there are different levels that are hierarchical. We can think of this in terms of like a Venn diagram. We might have here all of the numbers. Inside of all of the numbers, we might have here all the real numbers. Inside of the real numbers, we might have a subset of the real numbers, and the subset is going to be the rationals, things that can be divided. So rationals get a Q for quotient usually. And here we can see if something is rational, it's also a real number, and it's also a number. So if we call something a number, that's not enough to know you can write it in the form of A over B like a rational. But if you know you can write it in the form of A over B, well, you know it's a real number and you know it's irrational. So the broadest category is the domain. There are only 3 domains of life. Those 3 domains of life are the archaea, the oldest, the bacteria, which are all single-celled, prokaryotic, and the eukarya, which can be single-celled or multicellular, and they have a nucleus, so they have a cell with a nucleus in the middle. The nucleus has a membrane holding the DNA. Karya is talking about kernel, and the kernel is what's holding our DNA. So we have a membrane wrapped around our DNA. That's special in the domain eukarya. We are eukaryons, fungus are eukaryons, plants are eukaryons. Pretty much everything that you would think of as alive is eukarya. Bacteria, you have to be thinking kind of specially. In archaea, most people don't think about it all. Species is the most specific level. If we're looking at the species Homo sapiens, there's just one species Homo sapiens. If we're looking at the species Gulo Gulo, there's only one of those. But inside of the domain eukarya, there are lots and lots of species, millions. After domain, there's kingdom, which is broader, like less broad, but still pretty broad. Then phylum, class, order, family, genus, species. This is a thing we need to memorize in order. And you may want to make some kind of mnemonic device. The best ones are not ones that I can put in a recording, but you'll figure out your own. So this slide here is looking at maybe classification of primates, and we want to look at the hierarchical nature of it. So here we're looking at apes, Old World monkeys, New World monkeys, tarsiers, lorises, and lemurs. They're all primates. Inside of the apes, we have gibbons, gorillas, chimps, bonobos, humans, and orangutans. So if we're looking at humans, we are definitely apes, just like chimpanzees, gibbons, orangutans, etc. But if we're looking at apes, if we know something is an ape, that doesn't mean we know it's a human. It could be lots of different things. So we're looking at like subsets if you're studying set theory in math, maybe. Sets and subsets. So examples here, all humans are apes, but not all apes are humans. All apes are primates, but not all primates are apes. So here, this is all primates that splits off into lots of things. Apes is one of them. If you know you're an ape, then you know you are also a primate. But if you know you're a primate, you could be a lemur. All primates are mammals, but not all mammals are primates. So all the primates are mammals. This isn't on the chart, but a horse, a cat, a dog, whatever, those are all mammals too, but they are not primates. If we're looking at mammals, which is way down the chart here, they're way down there somewhere. Mammals is broad. If you're saying mammal, you could be talking about a cat, a dog, a horse, a duck-billed platypus. Or you could be talking about a primate, like a lemur or a monkey or an ape. But if we're looking here at orangutan or human or bonobo, these are individual species. There is no subcategory. In humans, if you're a human, you're a human. There's no subcategory beyond there. We need to know about the 3 domains of life. The archaea are the oldest, we think. They are prokaryotic, means they don't have their DNA in a nucleus. Single-celled. Just like bacteria, most bacteria have flagella. And we are in the domain eukarya. We are eukaryotic. We have a membrane around our nucleus. We could be single-celled or multicelled. I myself am multicellular. And all eukaryotes reproduce sexually. This is like requiring a disclaimer here, can reproduce sexually. We need to know how to make a dichotomous key. The purpose of a dichotomous key is if you find some species, some example of a plant or an animal or whatever, and you want to know what it is, it's impossible to memorize them all. There are too many. So instead we have these tools called keys that we can use to figure it out. A key is something that is difficult to make. We're going to learn just the basic idea of how to make it. But the idea of the key is somebody should be able to look at something. You give them a list of questions without knowing what they're looking at. They should be able to answer those questions by looking at their thing and answering as truthfully as they can and come to the right name. So some similarities people have said, this is like Choose Your Own Adventure, those kind of books. Or there's a game, Guess Who. If you don't know it, go look at a YouTube version of it or something like that. It's basically the same idea. When we're making a dichotomous key, you'll need to be able to make a series of questions that anybody could read to identify what you're looking at. So if you have these leaves available and you find this one today and you say, what is it? You go look at your tool, which is the key. You should be able to answer the questions about this leaf and figure out that this one is a birch leaf. So is this a compound or simple leaf? We can look at the definition, but this is a simple leaf, so we go to step 4. Step 4, do the veins branch out from one central point or off a main one in the middle? Looks like off a main one in the middle. Go to step 6. Step 6, is the edge smooth or toothed? It is toothed, so it's a birch leaf. And we should be able to, no matter which one we look at, if we started by looking at this one, we should be able to get the right name for it. Or this one, we should be able to get the right name for it. This same set of questions should get us the right answer for anything in this group that we ask about. Here, we're kind of taking a step into the second half of Unit 6. In the second half of Unit 6, we're looking at how populations or species change over time. So we're getting into the idea of evolution. When we reproduce, our offspring are not always exactly the same. In fact, they are almost never exactly the same, even if we reproduce clonally by mitosis or something. So every time that we reproduce, we have some small amount of change in our DNA. The copying of DNA is a complex process. We do it very well, we being things with cells, things that are alive, but that doesn't mean perfectly. So there will always be some random changes, we call those mutations. In things that reproduce sexually, then those changes happen more quickly. There are things that can increase the rate of mutation, things like various chemicals or UV light, other types of radiation. Most changes in your DNA don't help. Most of them are harmful, or if not harmful, don't have any noticeable effect. Sometimes they turn out to be helpful. Rarely, but sometimes. And that's what's really important. We are looking at, whenever we're looking at things that are alive today, the descendants of the lucky ones, the ones who had lucky beneficial mutations, they're the ones who did better than their competitors. And they're the ones who had more offspring, and we are their descendants. Well, and so are all of the things we see alive today. The elm tree is the descendant of the elm tree that was the luckiest. The changes that we see as a result of these mutations or sexual recombination are called variations. Variations just mean differences, doesn't mean good or bad. If a variation is beneficial, which we define as, does it help you survive and reproduce? Do you have more offspring, more healthy offspring, than somebody who doesn't have that variation? That's it. That's the definition. That's what we can say is good. Most changes, most variations do not help, but sometimes they do. And if they do, because of the definition, which means you have had more healthy offspring, that means the population is going to look more like you in the future. If you have a variation that lets you survive a disease that is currently ravaging your population, that's great news for you and for your offspring, and your offspring are probably going to be alive while other ones die. If this changes over many, many generations, then the whole population starts to look different. How these changes appear in a population is a result of what's called natural selection. This happens over a long period of time. This is many generations. This is something that is not happening on one day. It's not happening to one individual or even one small group of individuals, but over a long, long period of time over the whole population. Doesn't happen to the individual. Individuals, they just live and die. They have offspring or they don't. That's not the same. It's an important part of natural selection, but it's not all of it. Even if you have a beneficial variation, something we call an adaptation, that doesn't mean that you're done with that. That may still have some new variation to it. And that might be undoing something that's good. That's a total possibility. For the variations that are beneficial, the adaptations, they need to already be there before we find out if they're good or not. The change happens, and then it gets tested when the organism is alive. This is a very, 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 very commonly misinterpreted thing. For natural selection, we can never say, oh, this organism changed, this species changed because of, or so that they could. No. There was a change, and the result was either beneficial or not, but they never had a plan like, oh, you know what I would really like is I'd like to be able to do photosynthesis. No, nobody knows what that is until it happens. You don't even know if it's beneficial or not until you try it. Two ways to explain this. Wallace's theory. So Charles Darwin and Alfred Russel Wallace both had similar ideas at the same time. Darwin gets in all the books, though. Their idea is the one we're going to learn because that's what we've based all of our modern understanding of evolution off of. Of course, we have changed details as we learn more and more, but the basic idea is still good. Lamarck we learn basically as a counterexample, one way to watch out for how we think. So, Lamarck's idea was called inheritance of acquired characteristics, which means that the changes you experience in your life can be passed down to your children. And this is almost like the right idea, because changes in an organism do get passed on sometimes to the next generation, but not changes that happen to your somatic cells. Somatic cells means all of the cells of your body that are not gametes. Gametes, which are sperm and egg, these are the ones that are carrying the pieces of DNA that will actually be inherited from one generation to the next. Darwin didn't know this. Lamarck didn't know this. Wallace didn't know this. Nobody knew it for a long time yet. But the only thing that actually gets transmitted from generation to generation is the DNA that's in the gametes. So only changes to the gametes can affect the next generation. Lamarck's idea doesn't work because if you do a lot of exercise, you get very fit. That does not mean that your children will be very fit. They'll have to do their own exercise. If you pass Bio 20, that doesn't mean your children know everything from Bio 20. They're going to have to learn it again on their own, hopefully with newer and better information. So Lamarck's idea was changes in your lifetime could be passed on, and that doesn't work because they affect your somatic cells. Darwin's idea was there are changes that do get passed down, but not changes that you can make yourself. So Darwin, or Wallace's idea, was that he noticed there are variations caused by mutations. He didn't know that, but he noticed that there were differences. If you look at every human or every turtle or every whatever, everyone's going to be a little bit different from the other. Some of those variations are beneficial, some are not. Most are not. This is a really key idea. Not every organism survives to reproduce. That's true for any population. Whether it's humans, whether it's bacteria, it doesn't matter. The number that are living, that are born, is not the same as the number that survive until the reproductive age and successfully have offspring. This is true for everything. So this is our filtering mechanism. If you didn't survive that long, then you don't get a chance to reproduce. And only the ones with a chance to reproduce do reproduce. So the ones that do survive to reproductive age probably have something going for them that's better than the ones that didn't survive. Over many, many, many generations, sometimes thousands, tens of thousands, hundreds of thousands of years, those adaptations will spread throughout the population, and they'll just become normal. That's just what butterflies look like now. That's just what beetles look like now. The difference between the ones that survive to reproduce and the ones that don't, we could say it's their different variations or adaptations or not. But what we can say is they have a selective advantage, which means that they have something about them that allows them to be selected to survive or not selected to die. The selection sounds like an active process, but this selection is done by nature. It could be done by temperature. It could be done by climate. It could be done by pH. It could be done by predators. If we do this ourselves, if we do artificial selection, which we do, we do it a lot, we could be selecting for only the cutest dogs, and we end up with tiny little cute dogs that look like puppies their whole life. We've done this. We could be selecting for only dogs that are the best at hunting rabbits, and we get ones that are really good at hunting rabbits, and we've done this. We could select for dogs that are really active and fast, or dogs that are really slow, and we've done this. The way we do it is the ones that have the characteristics we like, we allow to reproduce, and the others we don't. Nature doesn't, as far as we know, have any kind of preferences, but the same basic process is happening. Some things survive and others don't. So if nature is doing it, we call it natural selection. If humans are choosing, we call it artificial selection. But it's really the same process. The ones that get to reproduce are the ones that get to pass on their DNA. In terms of evidence of evolution, we need to understand a few different ways that we have come to see this process happening, even though it's happening very slowly, not something that we can witness really in a human lifespan. We still have some evidence that it is going on. One of the best ones is looking at the fossil record. If we look at what is buried in the ground, the bones of things or records of things that are buried in the ground, the patterns from deeper down where things are older to closer to the surface where things are newer, those patterns more or less match across the earth. The more or less comes from different plate tectonic actions. And there is a general pattern for the types of things we find from 1 billion years ago to the things we find 100 million years ago to the things we find 1,000 years ago, etc. Fossils are great, but rare, very, very rare. So there are a special category of fossils called transitional fossils that we really love to see because they answer some questions for us. Like, how did things start flying? And the Archaeopteryx was a great one of these because it's like a dinosaur, but also like a bird. Or Tiktaalik, which we found up in Nunavut. Sort of like a fish and sort of like a lizard, sort of halfway in the water, halfway out of the water. And helps us to identify how these big steps could have happened. Because that's always been a question. Okay, sure, small steps everybody can see, but for big steps like starting to fly or coming out of the sea, that's huge. How does that actually go? And these ones give us some clues. If we look at where fossils are found in the world, we can find fossils in, for example, Brazil and the Ivory Coast that are of very closely related organisms, even though they're an ocean apart. And that only makes sense if they were alive at a time when Brazil and Africa were touching each other as they were. They're currently moving away from each other at a very slow rate, but give it enough time, and you hit a whole Atlantic Ocean in there. 2 words that we need to know for sure are homologous and analogous structures. Homologous structures are things that maybe they look different, maybe they even do a different job. But if you look at how they're built from the same parts according to the same plan. If you look at a human arm and a horse leg and a whale fin and a bat wing, you might think they're very different. But if you look at the bone structure, you notice, hey, it's got the same number of bones in the same orientation. That's weird. They must be variations on one plan, one set of DNA instructions. This is evidence that things are related, that we otherwise might not guess. Analogous structures don't do the same job. It's not evidence that things are related. Analogous structures are things that look similar, but it's superficial. If you look at a bird wing and a fly wing, they both are wings. They both let the thing fly. But a bird wing has bones and blood and muscle, and a fly wing is made of chitin and it has nothing like muscle. Very, very different origin, which doesn't mean that they are closely related at all, even though they look kind of similar. We'll talk about embryology in class, looking at how embryos develop through their gestation. This was a really clever observation. Started us thinking about a lot of evolutionary relationships between things, but it's just not really good. We don't use it really anymore at all, but it's an important one in the history of starting to understand this concept of evolution. For genetics, we're looking at if you take a gray cat and a white cat and you mate them, what color cat do you get? If you take this kind of bean and that kind of bean and you crossbreed them, what kind of bean do you get? In Bio30, you're going to do lots of examples of looking at genetics, and in particular, you're going to do crosses in things called Punnett squares. There's a little bit of math coming in your future. It's easy math, but there it is. This one here, molecular biology, is the best piece of evidence that we have for evolution these days. Because DNA is the molecule that is transferred by our gametes, by our sperm and egg, to the next generation. Changes in that DNA are directly related to what the ancestors looked like and what the descendants will look like. So if your DNA molecule is more similar to another organism's DNA, there's only one explanation for that. That explanation is you have a common ancestor. This is by far the most reliable, just because of, frankly, just because of math. The number of molecules, number of atoms, number of individual pieces in a DNA molecule is so high that we can very confidently, just using statistics, tell how closely some things are related. The chances of DNA molecules being similar to one another just by chance and not actually related are so low that we can almost, and we do, ignore them. The chance that the DNA of two organisms will be the same, but they're not related, is basically zero. So if your DNA is similar, that means you have a relation. The closer your DNA is, the closer your relation. So when I say only one believable explanation here, I mean just through statistics. The creation of a new species is called speciation. And in order to create a new species, there has to be some kind of barrier. It could be a physical barrier or it could be a biological barrier that keeps parts of one group separate. A physical barrier could be something like a river, a highway, a mountain, something that appears. It's hard to think about a mountain or a river appearing suddenly, but we're not talking about small time spans here. We're talking about a long time span. So we have population that gets split in half by a river, and this group over here and this group over here, they may end up in slightly different circumstances. And what works over here and what works over here, in group A and group B, might be different. And over a long enough period of time, group A and B might become separate species. They might not be able to breed anymore, interbreed anymore. Or the barriers could be biological. So even if A and B are together in the same area, there may be changes in parts of the group that make them even more different from other parts of the group. So if you start singing a different song and you're a bird, well, the birds who are expecting to hear their exact favorite song won't be happy to hear your song, and they won't mate with you. If you make a different stinky pheromone to attract all the other moths over, and those moths don't like that pheromone, well, they will not mate with you. If you were doing an amazing dance to attract your mate, but it's not the right dance, then they won't mate with you. And that might be something, it might be something like you're awake at night and they're awake in the day. It could be a lot of different things, including your genitalia don't match anymore. But it could be enough to keep you from interbreeding, which is going to keep the two populations separate. So population A and B might end up over a long enough period of time becoming different species. One example of this that's famous, and I really do hope you take some more time to read about this, is adaptive radiation. The famous examples here are Darwin's finches in the Galapagos Islands, fruit flies in the Hawaiian Islands. This is where we get one species. So let's do the finches. We have some islands, series of islands. A bird comes, a mating pair of birds come somehow, by accident, through a storm. We don't know how they got there. But they come here and there are no other birds. So their population explodes, right? They're doing great. But over time, there's a lot of them. There's no more room there. They have a meta-limiting factor there. So some of them maybe are better at eating nuts, and some of them maybe are better at eating insects, or some of them like to live up high or live down low, or some of them have some difference that lets them exploit some other niche. And that difference might keep group A in one area and keep group B in another area, and they might not interact very much. And group A and B might be separate enough that they only mate. A's only mate with A and B's only mate with B. And you might end up with different species over time. Adaptive radiation means we have one species that radiates into, radiate means starting at a central point, ending at many different points. So species A, maybe, here's the ancestors or descendants, of species A. But maybe there's also another group that becomes a whole new species, and another, and another. So this radiation means one species becomes many species. There are lots of examples of this in nature, but if you want to read about it, Darwin's finches are probably definitely the most well described of those. And how fast species change could be slowly, small changes kind of adding up over time. That's one idea called gradualism. And the other idea of a species mostly staying the same, and then suddenly there's a catastrophe. The nearby volcano erupts, or there's a flood, or a new competitor moves in, or something. And most of them die, but some of them who are a little bit different continue on. And now we have a new species, or maybe two differences make A and B different. A exploits one new circumstance and B exploits another new circumstance. So gradualism is like small changes adding up over time. Punctuated equilibrium is things kind of going along as normal until a disaster, which makes the living conditions for the original species less good. So the ones who have variations that weren't beneficial before, those variations might become beneficial now. And if that's the case, then they may, over a longer period of time, become a whole new species. So this is a quick overview of what's going on in Unit 6 and the last unit. Hopefully this